Hi, my name is Francesca Balestrieri, and this is my exam mini video 4. There are a lot of different proteins involved in DNA replication. I'll be using different shapes to represent these proteins and keep a running key telling you what the shapes represent on the right. DNA replication starts when an initiator protein binds to the origin of replication, which is represented by a yellow star. This causes local unwinding, which creates a short stretch of single-stranded DNA. DNA is a plectonic coil, meaning it must be unwound before it can be pulled apart. To facilitate this unwinding, helicase attaches to the replication fork and moves along the length of the DNA, breaking the hydrogen bonds that are holding the two strands together as it goes. I'll be representing helicase with an orange triangle. As helicase is separating the DNA strands, single-stranded binding proteins, represented by gray circles, coat the now single-stranded DNA to protect and stabilize it. The single-stranded binding proteins also ensure that the single-stranded DNA doesn't bind to itself. The green arch represents a gyrase, which is a topoisomerase that aids in the unwinding process by releasing supercoiling ahead of the replication fork. From this point on, we're going to only look at one side of the replication bubble for simplicity's sake. Now is a good time to mention that replication will be occurring in different directions on each strand. DNA is synthesized from 5' to 3', so the template DNA must be read from 3' to 5'. Because of this, replication will proceed to the left on the top strand and to the right on the bottom strand. DNA polymerases are responsible for synthesizing new DNA, but they cannot do this from scratch, as they must have a free 3' OH that they can add a nucleotide to. To solve this problem, RNA primers are formed by Rna Primers. Here the RNA primers are shown in purple, and the primase is represented by a white cloud. Since the bottom strand is being replicated towards the origin of replication, new primers must be laid down every so often as helicase makes its way down the replication fork. Only one RNA primer is needed on the top strand, however. Once the RNA primers are laid down, DNA polymerase III can add nucleotides onto the primer. DNA pol III is represented here by pink arrows pointing in the direction of replication. DNA pol I is also shown here by a black box. DNA pol I has exonuclease activity that is responsible for replacing the RNA primers with DNA. If something happened and DNA pol I could no longer perform its exonuclease function, then there would still be RNA primers along the newly synthesized strand of DNA, which we do not want. Also, remember how I said that replication is going in different directions on each strand? Because of this, and the fact that the origin of replication is in the same place for each strand, one strand, the bottom one in this case, must be replicated discontinuously. We hinted at this when we mentioned that several RNA primers were laid down on the bottom strand. Along with this discontinuous replication, the replication on the bottom strand also lags behind the top strand. Because of this, the strand that's replicated discontinuously is called the lagging strand. The top strand, on the other hand, is replicated continuously away from the origin of replication. It's called the leading strand because it is able to be replicated before the strand that is replicated discontinuously. Now that DNA polymerase I has replaced the RNA primers with DNA, we can see that the discontinuous replication of the lagging strand has caused the DNA here to be split into fragments. These fragments are called Okazaki fragments. Ligase, represented by a blue heart, is then responsible for sealing the gaps in the sugar phosphate backbone. And that, my friends, is DNA replication.